Microorganisms underpin virtually all elemental cycling in aquatic ecosystems. In his foundational work here at Cedar Bog Lake, Minnesota, Raymond Lindeman developed his trophic dynamic theory of ecology, in which he placed bacteria and their associated detrital ooze at the very center of matter and energy recycling. Lindeman noted that he had been inspired by François-Alphonse Forel, who in his founding monograph in Limnology drew attention to the ubiquitous presence of bacteria and to their key roles in the aquatic carbon cycle. Today, microbial ecology is one of the most active areas of research in limnology and oceanography, addressing questions about biodiversity, biogeography, food webs, biogeochemical cycles, greenhouse gas fluxes, dissolved oxygen, and water quality. Microbial communities in the water are composed of numerous taxa of prokaryotes, eukaryotes, and viruses. The molecular revolution in our understanding of microbial biodiversity has taught us that the prokaryotic communities in Lindeman's detrital ooze actually included two very distinct domains of life, bacteria and archaea. Eukaryotic microbes in the water include photosynthetic protists, the algae, non-photosynthetic protists such as colorless flagellates and ciliates, and diverse microfungi that are still very poorly understood. However, the term microbes has often been used in aquatic ecology to refer more exclusively to bacteria and archaea. Microbiota refers to the assemblage of microbes, while the term microbiome is best defined as encompassing the whole microbial ecosystem. That is, the entire community of microbes, the microbiota, plus the genes and products of their collective activity, plus the surrounding environmental conditions that the microbes interact with. Microbial cell numbers in surface waters are generally around 10 to the 5 to 10 to the 6 cells per mil, and populations vary within this range among seasons, across productivity gradients, and with depth down the water column. Free-living bacterial cells can be very small, resulting in high surface-to-volume ratios. Some planktonic bacteria are motile through a variety of cellular mechanisms. These allow such cells to move through the water, which is highly viscous at this scale, to reduce diffusion gradients and to seek out chemical micropatches. Particle-associated bacteria depend on higher substrate concentrations, and many can move over surfaces, including by extension and contraction of protein filaments or pili. Bacteria have high affinity transport systems in their membranes that can deplete solutes in the surrounding environment to concentrations below detection limits. In systems that are phosphorus limited, for example, this high affinity transport machinery gives bacteria an advantage that allows them to compete fiercely with the algal plankton. Microbes drive overall detritus decomposition and element recycling, mineralizing both carbon and essential nutrient elements such as nitrogen, phosphorus, and sulfur. The study of elemental ratios and their influence on ecology and evolution, that is, ecological stoichiometry, has been widely applied in limnology, including in aquatic microbial ecology. And here is introduced by Dr. Jim Cotner, a professor in the College of Biological Sciences at the University of Minnesota, St. Paul.
Way back in the day when I was working with Bob Wetzel, who many of you may know or at least know of, I had a strong interest in phosphorus and bacteria. I continued looking at phosphorus and bacteria in my graduate work, and one of the things that I realized is that microbial phosphorus and carbon are very tightly coupled, which sort of led me down the path toward ecological stoichiometry. Going back to our work with Bob Sterner and Jim Elsevier, initially on the growth rate hypothesis. More recently, we have focused on a lot of microbial homeostasis, essentially whether or not bacteria are what they eat or whether or not they can maintain some sort of stoichiometric homeostasis, despite the fact that their food changes quite a bit? The answer, like most things, is that it can be both. Basically, there are different bacteria that do different things, but in a community of bacteria, it's pretty likely that there's some strain that is able to grow effectively, no matter how extreme the substrate conditions might be. In the figure in Wetzel's Limnology, adapted from the work that Casey Godwin did in my lab, we demonstrated that one bacterium could grow in a chemostat where we supplied a organic carbon to phosphorus ratio of over 200,000 to 1. Its biomass composition was not 200,000 to 1, but it was 10,000 to 1, 2 orders of magnitude greater than the Redfield ratio.
This diversity of responses to elemental ratios within and among communities also implies a large genetic diversity among bacterial species. Comprehensive inventories of this diversity have only been possible since the mid-1990s, when DNA-based tools became available. Microbial ecologists use a highly conserved genetic locus, the 16S rRNA gene, to identify and classify microbes. Application of this method has shown that freshwater microbial communities are incredibly diverse, with hundreds to thousands of species per milliliter of lake or river water. However, rank abundance curves show that often only 20 to 30 species constitute the large majority of each community, and that there is a long tail of rare taxa, sometimes called the rare biosphere. Many of the species considered part of the global freshwater microbiome are found exclusively in freshwater, and only a few taxa are found in both marine and inland waters. The most abundant taxa in lakes can persist throughout much of the year, and most have very small cell sizes with minimal genetic content. Taxa that favor high substrate availability, called copiotrophs, have boom and bust cycles that are therefore largely undersampled. In the classic aquatic food web model, primary producers are grazed by zooplankton, and carbon and energy then passes up the food chain to fish, with microbes acting as decomposers and recyclers. But in 1983, Farouk Azam and his colleagues proposed a more resolved conceptual model called the microbial loop, with bacteria and dissolved organic matter, DOM, at the center. DOM is released by phytoplankton and is taken up by bacteria, which are grazed by protists, in turn eaten by zooplankton, which are preyed upon by fish. Viral attack and lysis of phytoplankton and bacterial cells contribute to this DOM flux, and this viral shunt directs organic carbon back into the microbial loop. Most microbial ecologists would argue that freshwater microbial communities are determined largely by bottom-up factors. These include the availability of organic and inorganic substrates, as well as temperature, light, salinity, pH, and redox conditions. However, microbial communities are also controlled by top-down factors, primarily grazing by protists and lysis by viruses. Predation by eukaryotes, protists and metazoa, seems to have a larger effect on biomass, while viral lysis influences diversity, although at times can equal the impact of predation on biomass. Competitive interactions among pelagic microbes are greatly altered by grazing pressure. Microbial defenses to this predation include size reduction, filament formation, altered cell wall structures, exopolymer production, cell signaling defenses, toxin release, and altered motility patterns. Viruses are enormously abundant in aquatic environments. Concentrations of viral particles range from 10 to the 6 to 10 to the 8 per mil, and typically exceed bacterial concentrations by 1 or 2 orders of magnitude. Virus morphology can be observed using electron microscopy, and the resultant images hint at the large diversity in natural viral assemblages that is increasingly apparent from molecular studies. The most abundant viruses in lakes and rivers, as in other environments, are bacteriophages or phages, which parasitize bacteria. The enormity of bacterial lysis by phages and the selection for resistance against these attacks has resulted in strong coevolution between bacteria and their phages. Phages also contribute to genomic changes in microbes by acting as a vector of horizontal transfer of DNA, genes, from one microbe to another. The availability of genomic data from aquatic environments has already yielded many surprises that were invisible without these methods. Extreme aquatic environments, such as this ice-capped Meromictic lake in the high Arctic, have proven to be rich sources of unexpected taxonomic and functional diversity. Genomic analysis of this coastal lake called Lake A has shown that diverse taxa of bacteria, Archaea and eukaryotes, are segregated across environmental gradients down through the water column, and a new bacterial class was discovered in the deep, dark, sulfitic waters of the lake. The lake also contains diverse viruses, as described here by Dr. Thomas Pitot, a microbial ecologist at University Laval, with a special interest in giant viruses, a newly recognized component of aquatic microbial diversity.
Giant viruses are classified as nucleocytoviricoda, and the host cells are eukaryotes, such as algae and amoebae. They have many interesting features, including the large size up to 0.2 micrometers in length and very large genomes with numerous accessory genes to optimize their growth. Our work has shown that Lake A, other Arctic lakes, and similar lakes in Antarctica, such as Lake Frixell and Ace Lake, have distinct and rich assemblages of giant viruses. In Lake A, as for the other components of its microbiome, different taxa of these viruses occur at different depths across the environmental gradients of the lake.
These genomic approaches are also leading to new functional insights into lake ecosystems. For example, microbial DNA analysis has identified the widespread distribution of rhodopsins that may subsidize chlorophyll-driven production, the central role of nitrogen-rich DOM, and new intermediates in the biogeochemical sulfur cycle. These studies have also revealed a surprising number of freshwater genomes affiliated with taxa known to metabolize C1 compounds, such as methane. These include methane oxidizers, such as methylopumilus, that are highly abundant and prevalent in most lakes, affecting oxygen as well as carbon dynamics. Back to Dr. Jim Cotner, who describes his current work on microbial methane cycling and its potential impacts on water quality and on fish habitats.
The work we've been doing in the Cotner lab most recently is focused on greenhouse gases. We're focused on the big three, CO2, methane, and nitrous oxide, but our strongest emphasis by far of late is on methane because ponds, lakes, and wetlands are the primary natural sources of this gas to the atmosphere. We were able to secure funding from the NSF right before the pandemic to build these profiling buoys, which we have put in 3 different lakes. One of them is Cedar Bog, which of course is the lake that Lindemann did his famous work on the trophic dynamic aspect of ecology in. All of our work on greenhouse gases has shown that stratification is a key driver of methane production. That is, when lakes are stratified longer, more methane is produced and less of it is degraded before it can escape to the atmosphere. We've also come to realize that methane is a huge sink for dissolved oxygen, which means that it's a big issue for water quality. In Minnesota, we're proud of our lakes. and the fish that they produce. But many of the cold water species, such as walleye, cisco, and lake trout, are threatened by low oxygen availability in deeper, colder waters. Our work suggests that methane degradation might contribute to the problem, mostly because it's absolutely delicious for many of the microbes that can consume it.